Welcome to the Molecular Moments Podcast.
In today's episode, we sit down with our guest, Dr. Kevin Bateman, Scientific Associate Vice President at Merck. Kevin is a veteran of the bioanalytical industry. He's known for innovating on a level many of us never have the opportunity to do. He's always thinking outside the box, making bold moves to progress our industry forward. He brings unique insight, experience, and excellence to everything he does. He's the guy I look to when I wonder, what are we going to be doing in our labs in 5 or 10 years? I hope you'll enjoy our conversation. We're talking science as scientists do. So without further ado, here's another can't miss episode of Molecular Moments. Welcome to the podcast, Kevin. I'm delighted to have you join me today. And can we start with you just giving a few highlights from your career?
Yeah, great. Great to be here, Chad. Really appreciate the invitation to come and have a conversation with you. Yeah, some of the highlights is spending some time with you at various conferences.
Appreciate that.
Reality, this is my 25th year at Merck, right? And so, and that's a long time at one company, and I'm pretty lucky to have survived those many years in the pharma industry, right? Because there's been a lot of changes over the years. So, lucky enough to start my career in Canada, and then relocate down here to the U.S. So I'm at the West Point site just outside of Philly, and I don't know. It's been a fun journey, and the journey continues. So access to technology that never would have seen before. You know, when you think back 25 years and how it's constantly evolving. So for me, the highlights are the change, the constant change, and the push to get better over the years.
So Kevin, tell me about some of the different responsibilities that you've had at Merck over that 25-year career.
Yeah, it's been an interesting journey starting. Obviously, when you first start, you're in the lab, right? You're in the wet lab doing experiments at the bench. And as time goes by and you learn more and gain responsibilities. So in my first 13 years at Merck, it was in the lab, focused on, it was an analytical lab. So we did everything. We did PK, we did MedID, we did biomarker work. And my job was to make sure we had the right capabilities to do that. I worked with chemistry, I worked with pharmacology, I worked with biology. And then when I moved down here to the US, I moved into the regulated space, working on clinical trials and worked on method development and running a methods development group for LCMS assays for small molecules, and then moving into peptides and proteins. And around that time, I said, okay, doing protein mass spectrometry is an area that Merck needs to expand. So I stepped out of that role and took on a role to build a protein mass spec group. Just me and a postdoc to start, and over the next several years built it up to like almost a dozen people.
Around when was that? Because I'm curious, trying to track against where, when the industry started making that transition, saying, hey, we need to be looking at large molecules with LCMS. Yeah,
it was around 2012, right? So a decade ago it started that transition for Merck. And I got to say, for Merck, we're traditionally a small molecule pharma company, and so biologics was not a big part of our portfolio. Vaccines always was, but like monoclonal antibodies was not, as big at Merck as for other companies. So probably a little bit behind in terms of what other companies were doing using LCMS for proteins. But I think we've done a pretty good job of catching up and leading in some cases. So, and then thread all the way through that, going back 20 years, was just how do we collect data that we couldn't get in traditional approaches. And so I'm alluding to small volume sampling. And to me, that started when I was in early discovery and we were doing animal studies in mice. And mice are really small and you can't get a lot of sample from them. And so how do we do studies with mice and use really small volumes? So that's what we worked on. And I started using dried blood spots in animal studies way back, 20 years ago. And then over the years, I kept pushing on that technology. It was a thread throughout my career and taking it all the way to the clinic. And then trying to advance the technology, because cutting your finger and dripping blood on a piece of paper is not really a high performance analytical technique. And so looking at other options in volumetric sampling and devices, and we continue in that space and trying to push it into even other applications beyond traditional drug level monitoring.
Yeah, so let me rewind you back to the beginning there with the DBS. I think a lot of people would actually say that the paper you put out in maybe 2004 or something like that was kind of I'll call it a seminal DBS paper for bioanalysis, right? We know that dried blood spots had been around and in use for heel pricks, for neonatals, I think going back to early 70s, something like that. But what, as an innovator, to move your company into that line of thinking and then to publish that sort of seminal paper that's going to be challenged, quite frankly, on the utility of it. Tell me about that experience and how you as an individual drove that forward, because it really did start something in our industry.
So it's interesting how life experiences impact what you do. And so for me, I'll blame my daughter, right? So around that time when I first started at Merck, I had my wife had our first daughter, and this
Glad
was
to hear
in
that
Canada.
was with your wife also, Kevin?
She was involved, right? Yeah.
Right.
So, but in Canada, the public health nurse would come to your house, and part of that visit was doing a heel prick and collecting a blood sample for testing for inborn errors of metabolism. It's just routine health monitoring, right? And this was when I was still doing my PhD before I started at Merck, right? And then when I joined Merck, I started learning about doing PK studies in animals, and again, back to the mice. And it was like, back then, you had to use one mouse for every time point. And this was expensive, especially if you're using knockout mice. And so I remember in a project meeting, this was the big topic. And it's like, why can't we just use a drop of blood, right? And I'm like, why can't we just use a drop of blood? And I remembered the heel prick, and it's like, okay, dried blood spots. And that's sort of, those events coming together, the need to improve how we do the analytical, the experience of seeing dried blood sample collected and saying, okay, and I hired a summer intern and gave him the project to work on. And that's, that was who was on the paper. So an undergrad student and myself working on dried blood spots and just putting it out there, say, here's a technique that, is going to help reduce animal usage and could be applied to just routine PK screening. It's interesting. It took a long time after that for it to be, looked into, and it's had its ups and downs. But I think what we've seen through COVID and the pandemic is the need to decentralize clinical trials and use remote clinical trials. So techniques like at-home sample collection of blood samples is becoming more and more mainstream. And to think it started 20 years ago with a mouse PK study. Right?
Yeah, that's pretty interesting. And I recently had a conversation with Neil Spooner, of course, who you know well, and has worked in that space and done a lot to advance that space. And he also referenced your paper when we talked to him, and which was cool. And I said, hey, we're talking to Kevin soon. So it's nice when we make those connections. And one of the things, and we'll talk a lot about other innovations and other things you've done at Merck with new technology. But one of the things that came through that I was thinking about is when you work at Merck and you're innovating and you're doing new things, and you have the competitive space you have to work in, and you also have that sort of non-competitive, and you're a scientist, you want to share those results. How do you make that balance at Merck as a I'm going to call you a serial innovator in what you've done? You want to share it all, but you can't share it all, right? How does that work?
The question, why not just keep it a secret at Merck? Because it's an advantage for Merck, right? And
exactly.
Because coming up with something new and innovative and then making it available for everyone else is potentially giving up a competitive advantage.
Right.
And we do have those discussions internally about, hey, is this something that we should keep to ourselves? But I think my attitude and sort of the attitude is, well, if we thought it up, someone else could probably think it up too. And putting it out there makes it generally available versus someone else might want to patent that approach and block us from using it. So Merck's attitude is, we don't want to compete on tools. We want to compete on molecules, right? And so put the tools out there and let the best molecule win from a patient perspective, right? And so so I always, taking that attitude to heart. It's like, hey, if this is a new approach, it's more likely to be accepted if more people use it, especially if you're going to take a new approach all the way into clinical application and have data go to a regulatory agency with a new approach. You want many companies coming with that same type of data because it's more likely to be accepted. Right.
One of the things you do is you are frequently asked to speak on new technology, and I've seen, gosh, I don't know, maybe a dozen talks from you where it's like, hey, Kevin reviews 10 different technologies in dried blood spots or in patient-centric sampling, 10 new mass spec innovations. And tell me first, sort of, how do you find time? I've often thought, geez, how does he read all these papers and get all this information and pull this all together? Because what I do is I just look at Kevin's publications and presentations and go, hey, look at all these cool things that are going on. So
I don't do any real work, Dad.
Yeah.
I just play. Right?
Yeah.
No, well, okay. I've been lucky, opportunity, but you got to be prepared. The prepared mind has, so being able to jump on new opportunities when you see them, right, has sort of been what I've been able to do. Part of it too is I have no hobbies or outside life, right? And so like work has been my hobby. My wife will back that up, right? And so, I was lucky enough that she would stay home and raise the kids, and I was able to go into work. And I think part of it too is the culture of when I first joined Merck at Merck Frost. I finished my PhD. When you do your PhD, you're working all hours of the day. And then when I joined Merck Frost, it was the same thing. Everyone was there all hours of the day. So it's like, oh, that's how you work, right? And there was the focus on, how do we discover new drugs and get them onto market? Like that was the mindset, and everyone, focused on that mission. So anything you can do to improve that and add value was encouraged, right? Yeah. So that's over my career. It's like, how do we improve what we do? I'm never satisfied to accept the status quo, right? And so lucky enough that I've had management support to encourage me to constantly try to improve things. Not 100% goes perfectly well, and sometimes the status quo is good enough. And I'd rather try to push for something and be told it's not needed than not push at all, right?
Yeah, excellent. So let's think back again, maybe 10, 12 years, or I don't know, you can go back as far as you want. Tell me a prediction you made or something you investigated, spent a significant amount of time on that was that was absolutely the wrong call, and one that was absolutely the right call.
Oh, man. Wrong calls get embarrassing.
Yeah, that's what I'm trying to do. So let's.
Yeah. I don't know. I don't know that they're the wrong call. I think things that have taken a lot longer to come into routine use. We can go back to, dried blood spots. It's been like 20 years and we're still not routinely using them in clinical development. And I think it's the learning along the way. It's like, okay. And for dried blood spots, hey, we can measure a drug out of a dried blood spot. That's great, right. But the reality is, if you want to do it for PK, there's a lot of other information you need to know to do PK. And so when did the person take the drug, right? And when was the sample collected? So while being able to measure the drug out of a dried blood spot sample is probably relatively routine, putting in the context of PK is not, right? And I didn't realize that when I first started on that. And so that's why over the last, 5 or more years, it's been trying to identify technologies that allow us to put that drug measurement in the context of PK, right? And so fail to understand the context of the whole experiment. You start doing things like, hey, I've delivered a solution for you. And it's like, well, actually, no, you haven't, right?
Yeah, Neil and I talked about that a little bit with regards to dried blood spots, because it felt like for a while it was the tail wagging the dog, right? With the dog being the clinical engine and trying to move these studies forward. And here we are as bioanalytical scientists, exactly as you said, saying, hey, we've got a solution. And they're going, we're not sure we have a problem.
Yeah. Well, it's finding the right use cases too, right? It's not a panacea. When does this make sense? And I think at Merck, some of the successes we've had are in things like episodic disease states like migraine, right? And so asking someone to sit in a clinical site and wait until you have a migraine before we take a blood sample is not really practical. But sending someone home and saying, okay, when you have a migraine, take the medicine, and then a little while later, take a blood sample, because then that makes sense. But just doing it because you can doesn't make sense.
I recently during COVID had, when you mentioned, pricking your finger and dripping it on a card and whatnot, during COVID, I was working with a company and we were doing some blood spotting. And for the first time, I pricked my finger and dripped it on a card. And it actually was difficult. Yeah. I don't want to I kind of figured it out, but it took some tries and things like that. And that's where I think evolving these technologies is so important past prick your finger and it's just going to drip. It doesn't really work that way.
No.
So, yeah.
And that's why, we've focused and moved away from that and gone to more of a device-based approach that has it all integrated to collect a robust.
So Kevin, as I mentioned, and as you mentioned, you've worked on so much more than dried blood spots and in the patient-centric sampling space. I want to hear about some of the really cool innovations that are going on right now. What I said in the intro that, hey, Kevin's the guy you ask when you wonder what you're going to be doing in 5 or 10 years. So tell me about, 2, 3, 4, whatever the really cool things are that you're looking at, that you're excited about for the future.
Yeah, there's a few. So one is just speed related to analysis, right? And so the last few years, we've invested in technology around really rapid mass spec analysis. So you can imagine, and I've looked at this many different techniques over the years, but most recently partnering with SciEx on their acoustic ejection mass spectrometry. So pretty much a second a sample into a mass spectrometer, so way faster than we could have done previously, right? So what does that speed mean, and how does that change our approach to experiments? And we're still in early days of doing this, and we've applied it, in other parts of Merck for high throughput screening, where it makes sense. You want to screen a library, and you have maybe you're looking at an enzymatic reaction where you put in a substrate and get a product, and perfect, mass shift, perfect for mass spec, right? But we've also looked at doing PK studies and what does it look like, and can we do quantitation, and we've shown that. And okay, speed maybe not the driver there for running PK, but there's a lot of experiments that you can do, and a lot of experiments you don't have to do because there's no chromatography, right? So you don't have to worry about changes in chromatography over time. The sample prep becomes much more straightforward, right? It just a balance between, do you get the sensitivity that you need to meet the assay criteria? But it also opens up completely, not new, but old ways of doing quantitation that we've not thought about. Things like standard addition, where you divide your unknown sample up into multiple aliquots and spike in known amount of drug into each of those and then run them. We don't do that because it takes a lot more analysis time to do that. And now with speed, we have the potential to think about new ways of doing quantitation, and how do we leverage the data sets where we could analyze samples multiple times and really understand the variability associated with them. So instead of using arbitrary guidance related, cutoffs on my accuracy and precision, we can actually truly measure the precision of our unknown samples and use that, which then goes into how we model the data too, and putting error bounds on the model. So you have to think about completely new ways of generating data and how that would impact our interpretation of those results. And really, at the end of the day, it's a better understanding of our drugs that we're developing. And if we can have data sets that help that, then that's what we should aim for. Right?
So yeah, that's really interesting. I guess I hadn't thought about it, because initially when I think about the technique, I think of, okay, we're not having internal standards, and so we're losing a little bit of control. So we have to get the technique really precise. But with standard addition, you could run multiple samples faster than you run one traditionally with less prep. And then you build the precision into the actual instrument analysis side of it. Then you got to shift that paradigm of thinking. And what do you, so let's say you're ready The next step beyond high throughput screening is probably to look at early discovery studies to get some quantitation, and then non-clinical, and then moving slowly towards clinical and IND enabling studies. What's your approach to moving people's thinking and ultimately the FDA's thinking on really overhaul? If that moved forward, you'd really have to overhaul, the 4615, 4620 thinking of acceptance criteria in studies. So how does one even start there?
Data, right?
Yeah.
Right, because that data talks and BS walks, right? Is sort of my view of the world, right? And so let's get the data. Let's let the data speak for itself, right? We're a science-driven, organization. We're a science-driven industry, and so if the data is compelling, maybe that's an approach that is worth implementing. Right? Yeah, and so we have to start early on. We can start with animal studies and show the value, right? And so this is me just spouting off, right? I have to
that's
prove
what you're
the value,
here for.
right? Yeah, and to me, I prefer the challenge, right? Hey, we've not done this, and I haven't thought through all of, the variables associated with it. And then that's sort of my approach of things. I jump in both feet and hopefully don't drown, and find people that are like-minded yet skeptical, right? Which is always why I've enjoyed talking to you, because you're like, okay, that's good. But what about this? What about that?
Right.
And I love those conversations, right? I rather have a good scientific debate with someone that is like, that's interesting, but you haven't thought it through, because that's where I learn, versus people that are like, okay, go ahead. I don't have anything to say or no, right? If you're going to push back on something I want to try, you better have suggestions on how to prove it, right? And so I think I'm going to go on a sidebar for a second.
Please.
But Merck, over the years have sends people on a lot of training courses and things like that. And I only remember one thing that I learned was about communication, and it's called the LCS model, right? Anytime someone has a suggestion or is putting out an idea, you should say one thing that you like about it, one concern you have about it, and one suggestion for improving it, right? And so if you can't do all those three, then you probably shouldn't be talking, right? All right.
I like that.
So, yeah. And so
it's good. It's good to know how the Merck training budget is spent. Kevin learned one thing in 25 years.
They gave up on sending me for training.
I suppose probably. but I think that kind of illustrates a point. Also, you're not concerned about saying what's on your mind, right? And I think people recognize where you're serious and where you're making a point, but still somewhat serious. And I think that's actually a communication skill that you have. So maybe you learned that in one of the courses and didn't realize it. But
we all need a bit of prodding now, right?
I feel like we've lost that opportunity that we get in conferences face to face and sitting down. And quite frankly, it's the sitting at the bar after a long day discussing the interesting presentations where that, for the people who aren't scientists that might be listening, that's what we do at those conferences, right? We talk about more science when we have a beer or a glass of wine in hand.
That way, really.
Yeah, kind of. But that's what we love. So a good colleague of mine, I've been giving her a hard time, but she recently said LCMS is really boring, Chad. And I said thank you very much. That's my almost 30-year career is LCMS. So. But it's all good.
There are aspects of it that are pretty boring, but I think that's a testament to the success of the technique, right? Yeah. It's if it's become so routine and accepted that it's become boring, then we are successful, right? But I think there's lots of other opportunity to make it exciting, right? That still need to be worked on.
Yeah, completely agree. So what else? Tell me about another innovation that we can discuss.
Yeah, so one that's tied to high throughput mass spec is, and then even in other areas too, is the rise in data science and big data and using large data sets to drive better decisions. And I think of it from an analytical perspective. The goal is give me a structure and I'll tell you the method before I even inject a sample, right? It's like, you think of all the methods that have been developed and validated and documented over the years, and we never leverage any of that data, which is kind of sad in itself. But imagine if we were able to capture that data and then use a structure to say, okay, in my huge database of previous methods, what's as close, to this molecule, give me a suggested sample prep, a suggested chromatography method, and also, predict the fragmentation and tell me what the method's going to be, right? We can't do that today, but there's no reason why we can't start building those models. There's already software for predicting chromatography out there, but, I think from a bioanalytical perspective, it's an untapped area that, potentially we could leverage. We're starting to look at and just mining the literature and using natural language processing to see if we can't pull out all the papers in the literature and look at the standard things that we talk about, accuracy and precision, and that are written into papers because there's been so many published that meet FDA criteria. Can we mine those, build us a database that then it's not to replace the analyst, it's to provide them a toolbox that would accelerate the process of method development, right? So that's one. Related to that too, because I've ventured into ligand binding land as well, away from mass spectrometry, and collaborated with people in that group. Part of the early process of developing a ligand binding assay is, especially for a therapeutic antibody, is to screen the antitype antibodies that you have.
Sure.
And so initially you might have 20 or 30 that you do a standard screening with that is, pretty routine these days, right. But you get down to maybe, 5 or 6 that you want to follow up on, and then you have to label them and do pairwise comparisons. And that becomes a very big experiment. And so we've worked at automating that and using some data tools to explore a much bigger space of, bigger experimental space. So assay formats, pairwise comparison, interference checking. I have a paper that is just going through internal clearance that sort of describes that approach. And we've been working with Purdue University and some of their data scientists to work on the data processing, and we're developing and making more data sets to allow us to, because the goal is to get a good pair of reagents to use and then take those into a full method development where we use things like DOE. But if you don't have a good pair to start with, then your assay becomes much harder to develop and validate. So
and it's expensive to pick multiple pairs. And it's time consuming to have to go back if you don't pick right the first time. And so I think these are the kinds of tools that we're going to need to really accelerate what we can do. So that's super cool. But I'm excited. Honestly, I'm excited for that paper. It's funny you mentioned, hey, I'm the mass spec guy. I'm now looking at ligand binding things, and I think that's I think I'm the same. Yeah, exactly. So it's an evolution in our industry. I think there's crossover everywhere where we used to have those kind of stricter lines of I'm a mass spec, or I'm a binding assay, or I'm a small molecule, I'm a large molecule person. So it's fun to have us all working together, maybe a little more than we used to.
Yeah, and then the thing that ties it all together that we have to consider too is automation and small volume dispensing technologies. And so we've used some, but we've recently invested in more automation and dispensing technologies to try to miniaturize assays, just to enable exploring many more experimental parameters, right? Because reagents are precious. If you don't want to waste them in using maybe traditional large volume approaches. So if you can miniaturize a lot of the experiments you do, then it allows you to do a lot more with the same amount of reagent and explore a bigger space. So, we haven't, 96 well plate standard format, but now, looking at 384 well plate formats and miniaturizing assay volumes, even for traditional assays like metabolic stability, PK, there's, certain others in the industry have gone to 384 well plate, but coupling 384 well plate with high content data capture, like on high res based systems, and data mining and marrying all those together is sort of an area that, for me is of interest and pushing forward is like, okay, combining all of those and not just keeping them as individual experiments, but thinking about how do we leverage all that data to make better decisions about
right.
not only the analytical aspect, but help drive programs along as well.
Yeah, and that's ultimately what we're trying to do. Another area I'm curious for your opinion on is where we're at with intact protein analysis by mass spec, and also I'm curious. I just saw the news release that Waters had acquired. I think the company's Mega Dalton. That was Dave Clemmer and Martin Gerald's company, and so that's even looking at intact viruses and capsids. And so, what do you think is the future there in that space, that really big molecule analysis?
Yeah, it's going to come down to what the use case is too, right? Because we've done some work just like other industry peers on doing PK of intact molecules in the preclinical space. It's a challenge, right? Because the molecules are big. They don't behave well from a chromatography perspective. The mass spec part of it is complex because of the big ion envelope and resolving power, but it's doable. And it gets down to what's the question you're trying to answer at the end of the day. Maybe early in discovery, you're trying to figure out, maybe it's you're worried about clipping, and you have like whether it's maybe an ADC or you have a protein construct that has a peptide attached, to an FC chain of an antibody.
Right.
And, you're worried about, does it get chewed up? Does it stay intact? Then using intact type of analysis is going to really help understand what you're measuring. Because if you digest it down to the peptides, you don't know if something had changed in vivo that you're missing, right? The virus stuff is pretty cool. Merck obviously works in the vaccine space, and probably is not in my area, but talking to the people that work there, like viral capsid full or empty?
Yeah, simple question, right?
Simple question, right? Complex analytical challenge. So things like virus detection, mass spectrometry is helping answer those types of questions. So it's pretty cool. Just again, expanding the types of applications that mass spectrometry can be applied to. So, pushing into higher molecular weight intact molecules from analysis perspective, but then applying native mass spectrometry to understand biologically relevant, folding or unfolding and or binding of target molecules to a protein. So all of these are, helping us understand better the biology of what's going on. So coupling that with things like cryo-EM and combining the data sets to get better insights into the structure of those proteins they're going to help, because that's the biggest challenge in pharma is understanding biology, right? At the end of the day, why do drugs fail? They fail mainly for lack of efficacy or for safety. And both of those are directly linked to our understanding of human biology, right? And so the better we can do it to help us understand human biology, the better and more successful we will be at bringing efficacious and safe treatments for patients.
Yeah, I'm just super excited by all this, Kevin. I really am. I think that's why we do it. That's why you said work is your hobby and reading papers and things like that, because we love it. That's why we stay late at conferences and go to the bar and talk science instead of baseball. Although sometimes it's baseball, sometimes other sports, hockey. Hockey with the Canadians, of course. But I was thinking actually just of. I always talk baseball with Brad Ackerman, of course, because he's also a Tigers fan. But just to throw another industry guy out there that I see as a great innovator and probably somebody I should do a podcast with. But anyways, I wanted to ask you, one of the things that you mentioned going through here was working with interns, with the new scientists and things like that. And I was thinking, as we're going through this conversation, we're sort of the first generation, you and I and other people who started their career in the 90s, kind of the mass spec applying bioanalytical chemists, right? And now we have this new generation, and I see across different meetings promoting young scientists and making sure that group comes along and has the same opportunities we did. So Tell me what you're doing and to try and make that happen for people and leave, I guess I'll say leave a legacy, although I think you're going to be around for quite a while, I hope, unless you won a lottery ticket or something and are looking to retire soon, but I don't see that happen.
Yeah. So you mean like, how are we attracting new talent?
Yeah, attracting new talent, getting them into bioanalysis, bringing them along so they have the same curiosity and desire to work long hours because they love bioanalysis, not because the company's making them do it.
I think one of the things that energizes me and that I've missed through the pandemic is actually going out to conferences and meeting and talking to students, in the poster sessions, and they're there. It might be their first conference, and they're so super excited about that. And to me, that's like super energizing for me, because sometimes we forget about what we do is actually, a pretty solid mission in life, right? We're helping advance human health. Sometimes we're so close to it, we forget and take for granted what we do, and we lose some of the excitement. So yeah, going and meeting students and talking to them, luckily at Merck, we have pretty solid intern program where we hire interns. We also have a postdoc program as well. And so that's good. We just got a new postdoc approved. One of the guys that report to me, we're going to have a postdoc come in and work on the high throughput mass spec project, right? So anyone out there listening that wants to do a postdoc, look me up, because
fantastic.
It'll be a good one. But we also have recruiting with universities, and we have a research center developed with Purdue University, where we do a lot of projects. Yeah, but then how do you, once they're here at Merck, how do we keep them energized and excited? It's a constant challenge because, we're dinosaurs, right? And I have two daughters, 25 and 23, and so talking to them and their attitude towards work and life and work-life balance is pretty indicative of people that we hire here, and it is a different attitude, right? It's like
it is.
I work to live versus live to work, right? I'm a live to work guy, but a lot of people prefer a work-life balance and come in and do really good work when they're here, but then they have a life outside of work, and I completely respect that. So that's just the balance of life, and that's just not me, but I fully get and understand how, like, my daughter is like, the clock hits 5, she's done. She does a great job working. She's an engineer and does a great job before that, but her life outside of work is equally or more important to her than her work life, right? And so
and I respect that as well, right? So live to work versus love to work, right? I mean, I think it's kind of I think those are tied together, and whether it's generational or something else, I don't really know, but they probably have it figured out more than we do.
But I don't know. For me, like just walking down the hall and talking to the scientists is, I find, one way of getting them energized, and because I don't know, I have crazy ideas all the time, and I'm like, hey, what if we did this? What do you think, right? And so it's kind of fun having those conversations with new scientists, and because they're not like, oh, that's just Bateman going off again. They don't know that yet. They haven't learned.
Right. Well, and you probably have to be careful that if you put an idea in their head and just say this or that doesn't necessarily mean run in the lab and spend a week doing it. Like we're just having the scientific. Oh, you want that? Okay. Especially if they're in somebody else's team and can do work to help you out.
Yeah, they quickly left the army. Yeah.
Yeah, that's fantastic. Kevin? You mentioned hockey, right? You're Canadian. It's almost you're almost born in your DNA. What other passions do you have outside of bioanalysis and mass spectrometry and innovation?
I got a great dog.
Yeah, well, that's good.
No. So so I guess you're asking what I do outside of work, right?
Yeah, what do you do outside of work, or maybe you don't?
Yeah, no, there's a few things that I've
You told me once you're lazy, but
I'm a pretty lazy, lie on the couch, eat bonbon guy.
Yeah.
No, so when the weather's better, I bike a lot, right? And so my wife and I, we bike. We've done a lot of biking trips all over, not all over the world, but in Europe and And that, I was never a big biker growing up, but when I moved here, I had my old bike from Canada, and there's good trails around here. So I started biking. And I said, okay, if I put 1,000 miles on this bike, I'll buy myself a new bike. And so I put 1,000 miles on it. So I went and bought a better bike. And then I biked on that for a while. And then we said, oh, let's go, a vacation, and we'll do a bike trip. And we went to Portugal. Actually, it was like tied. There was an EBF meeting in Lisbon.
Oh, right. Yeah, I remember that. Yeah.
And then right after that, we did a bike trip there. And it was harder than I thought. But it was so satisfying. And so when I came back, I joined a bike club, and I started biking more. And and so biking is my out on the road and doing whatever 30, 40, 50 miles is a nice mental break.
Yeah, fantastic. And it's a mental break, but also gives you a time to think and to give you white space to think about whatever you want, probably including science. So
and then hiking with my dog, right? Mm-hmm. I got a yellow lab named Penny, and my phone. I have lots of pictures on my phone, as you know, but most of them now are just pictures of my dog. Right, one from out hiking, and we go all over. And I find a dog a very calming influence in my life. Right, like I could have a bad day, I come home and my dog still jumps on me and licks my face and is happy to see me. My wife is happy to see me too sometimes, not always, but
she doesn't lick your face. No. Well, Kevin,
even when I ask,
And any closing comments you want to share? Any science we missed, or that we ought to at least look up, or anything else you want to share with everyone before we close this out? And I've had a great time talking. Thank you.
Yeah. No, one last area that is ion mobility.
Yes, please. Yeah. So, let's talk about it.
It's been around a long time, and there's good advances. So, so full disclosure, I sit on the scientific advisory board of Mobile Ion, and so they're making their slim technology commercially available. It's pretty cool, like high resolution eye mobility separations on the front end of a QTOF at the moment. But you can imagine it could be expanded to other. And so again, making faster analysis with higher content is again that data stream, that data, right? And so to me, the science of being able to generate more data, more relevant data, and then coupling it with data science is like the future of our industry, right? The regulated side of stuff is always going to be tough, but it's like, man, we do all that work to get plasma samples out of patients, and we measure one thing, right? For PK, and there's such lost opportunity. So the last one is again on the sampling and expanding beyond PK. We've done a lot of work over the last couple of years on multi-omic analysis of dry blood samples. We've done proteomics, metabolomics, some lipidomics, whole exome sequencing, RNA-seq, and we're in the process of putting these techniques into a translational oncology study. So it's going to be pretty exciting to get those samples collected and see what we can learn about integrating large data sets in collected remotely or in a clinical study. So I think the next few years of that are going to be pretty interesting. We don't know what is going to come out of it. And part of that is overcoming a risk aversion of, well, if we don't know, we shouldn't do it attitude, right? But we're not going to improve if we don't take some initial steps to collecting data sets that we've never had before, right? Some some may not add any value. I will fully, admit that. Others, we're going to gain insights that we could have never thought of before. Sometimes we don't like to do things unless we have a clear vision for what they're going to do, but sometimes we just have to explore and see where we land.
I love that. Explore and see where we land. I think that's a perfect place to leave it, Kevin. Thanks so much for all the insights. This fantastic. I hope a lot of people take the time to listen. Certainly, this was an episode for the scientists among the crowd because we really dug deep into it, which was fantastic. I loved it. Thanks so much. Kevin, that's all for this episode of Molecular Moments. If you enjoyed today's episode, be sure to subscribe to Apple Podcasts, Spotify, or on your favorite podcast app so you never miss a conversation. If you'd like to hang out with us outside of the podcast, we have many webinars and other presentations available for your enjoyment and education. Visit bioagilytix.com to see what's coming up and how you can stay in touch.
Thanks for listening to the Molecular Moments Podcast.